This is Immuno Africa, a podcast dedicated to spotlighting African immunology research.
The first edition of the Immunology in Africa podcast graduate student series seeks to spotlight the motivations, journeys, and perspectives of African graduate students in immunology while highlighting their amazing contributions to the field. It offers our listeners an opportunity to explore and keep tabs on the inspiring works of young African scientists in the early phases of their careers, especially those keen on strengthening the continent's capacity for scientific research, growth, and development. For this first series, we spoke with 6 graduate students pooled from 5 African countries whose research spans a rich diversity of viral diseases, vaccine development, cancer, obesity, autoimmunity, and allergies. The stories told here portray self-motivation, initiative, resilience, hard work, excellence, consistency, growth, discipline, planning, and patience. These stories offer valuable insights and lessons to young and emerging scientists interested in building a career in immunology, as well as established scientists in the field. The final episode on Immuno Africa Graduate Student Series features Kumba Seddu. Kumba started her PhD journey at Johns Hopkins University, where she investigated male and female immune differences to viral vaccines and infections. Her research added to the growing evidence that vaccinated females have greater antibody responses than males. This increased vaccine-induced antibody response in females is due to hormonal effects, estrogen, and sex chromosome effects, XX. Now at Harvard Medical School, she is studying the gene expression and immune cell activation cascade upon interleukin-2 stimulation. For her work, Kumba has won several awards, including the U.S. National Institute of Health Specialized Centers for Research Excellence Award, the Delta Omega Honorary Society in Public Health Award, and the Paul Farmer Honorary Award. Kumba is passionate about equipping researchers and young leaders from low and middle income countries to end infectious diseases and solve global challenges. This drives a commitment to mentoring students around the world and leading community solution-driven projects in our home country.
Well, first of all, thank you very much for having me. It is a great privilege to be on the podcast and share my story. So my name is Kumba Seddu, and I am from Sierra Leone, West Africa. So I was born there. I did most of my schooling there. And I then went to Singapore to complete the last 2 years of high school. And then I moved to the US to do my undergrad. And through that, I became interested in science and research as a way through which we can alleviate diseases. Going back, it's the desire to be a change in my community. So I grew up in Sierra Leone, as I mentioned, and seeing a lot of endemic infectious diseases and people dying from diseases that can be cured if there is like better research. So then I was like, okay, I was interested in medicine, but I was then thinking, so back in Sierra Leone, I was like, okay, medicine. So, and then I realized that, okay, in order to get the necessary training that I needed to leave the country to go to another place to like get the training and then eventually like give back, because Sierra Leone was affected by civil war. And so the infrastructure was not there. So then that was what caused me to start looking outside the country. And for my WAS, I had the best girls results for my WAS exam. And so that offered me a scholarship to go to Singapore. And then in Singapore, that is where I stumbled upon my research as a way through which you can study diseases. And so then that was when I was like, okay, this is something that I can be doing. So yeah.
Interesting. How did that interest in immunology research develop?
Right. So after Singapore, then I came to the US. And then in the US, I started doing a biology major. And during the biology major, I was still like, okay, very interested in research. So I reached out to a professor my sophomore year. So that is the second year for the US. Can I be in your lab? Can I do research in your lab? That was a neuroscience research. So even though infectious diseases was my main focus, but the initial opportunity for research was in a neuroscience lab. I took it because skills that are useful in neuroscience can be applicable to other fields of biology.
Yeah.
So even though initially I did not get the immunology. So I was like, okay, I can be trained here to think as a scientist, to be creative in the way I think, to be curious, to apply that. So I was in the neuroscience lab, and then that was during the 2014 Ebola outbreak in Sierra Leone, West Africa, that mainly
Yeah.
affected Sierra Leone. And there I was reminded again of how infectious diseases are plaguing the continent and Sierra Leone. So then I was like, okay, I need to find a way to like continue with that interest. So I took a class, but meanwhile I was still in neuroscience, the lab,
Mm-hmm.
learning how to become a scientist, learning the curiosity. And after taking the immunology class, so then I decided that post graduation, I was going to work in research before continuing to like graduate school. And so then that was when I moved to Boston to Harvard Medical School in the immunology department to begin the immunology journey. So that's how it started.
Yeah, yeah. I think I love how events kind of just several events just came together to just allow you pursue your passion and just feel your desire to pursue your passion, because you just highlighted the Ebola outbreak of 2014. I think it also affected Nigeria, maybe not as
Right,
severe as Sierra Leone was affected, but
right.
I think that experience in a way ignited your passion and made you feel like, yeah, neuroscience is something important, but yeah, something is going on in my country and I need to preserve this childhood dream and tackle infectious diseases
Right.
and in my setting. So yes, that's really an inspiring story already. And of course, how you also took the initiative to gain research experience as a
Right.
very young student in your second year. And yeah, so from Harvard, you started exploring immunology research more specifically, of course, from your moving from your background in the earlier research experiences you got in neuroscience. So yeah, so
Mm-hmm.
from Harvard, you gotten into grad school. That was like basically a research
Mm-hmm.
experience. So what has your research in immunology in grad school been like? So what have you been working on? And so just give us an overview of your work in grad school.
Right. Yeah, that's great. So yes, after Harvard, then I moved to Johns Hopkins, Bloomberg School of Public Health, to start my PhD there. And while I was there, some of the immunology work that I did was looking at viral infections and vaccination. So influenza and SARS-CoV-2, and particularly how males and female immune responses differ. So it was always known, right, that okay, males and females have different immune responses to several diseases, wherein for infectious diseases, males tend to have more severe outcomes, whereas for autoimmune diseases, females tend to have more prevalence of autoimmune diseases. But it was really the COVID pandemic that brought it to light, wherein now everyone was home and people were paying attention that males were getting sicker, and why is that? So then that really helped in bringing like the understanding to the forefront, not only in the scientific community, but also in general population.
Mm-hmm.
So I was looking at why this is the case, why males and females are responding differently. And it was a great opportunity back then during the COVID pandemic.
So what were some of the highlights of that research, and what are the implications? So why your findings, or why are some of your findings important? So just highlight some of your findings from your research and then highlight the relevance or implications.
Yeah. So the lab that I was in, some of the projects that I was involved in, some of the findings involved that post-vaccination. So this includes influenza or SARS-CoV-2. Females have greater antibody responses than males. So that is one. And another part too that the lab had shown is that this has to do a lot with hormones. So your level of circulating hormones, and particularly estrogen, is immunostimulatory, whereas testosterone is immunosuppressive. So that could be one way through which we're seeing that females have greater response. And the lab also showed that post-vaccination, female reports more adverse events. So adverse events doesn't mean that it is like bad. It's more like the redness, the soreness that someone can feel at the site of
Infection,
the
vaccination.
injection site. So yeah, so then it's female report more adverse events than males, and that is also due to like hormonal levels like estrogen. So because males that also had higher levels of estrogen had reports more adverse events. So those are some of the findings in some of the projects that I worked there at. And in terms of the implication, it has several implications. So for instance, I would say in terms of like vaccination, thinking about, okay, how can we tailor vaccines to boost male antibody responses similar to females, to make males like females? And it also has an implication in the sense that what about people that are going through hormone replacement therapy? So like people in like the menopausal age range, or people that are aged populations. So it has a variety of implications, and hopefully that work in the lab that they continue to do would impact how vaccination is done.
Yeah, thank you. Thank you for that summary of your work. So I think I just want to find out quickly whether you were working with just animal models or you actually worked with humans in the study.
Yeah. So yeah, for the projects that I work on, we're both animal and human.
Okay.
Yeah.
Okay. So I think still on your PhD journey, and you've been at Hopkins for a while, and you are moving, or you've moved to another amazing institution, Harvard University. So I think let's delve into the work you will be doing there, or the work you've done. I think you worked at Harvard for a while after you left your undergraduate study. So what will you be doing there now, moving to Harvard University to continue your graduate research?
Yeah. So continuing with my graduate research, so now I would be looking at how several immune cells in the body, like looking at their gene expression and regulatory profile. So like what genes are turned on, which genes are turned off in different contexts. And so, for instance, one of the contexts that I'm studying is in terms of stimulation with cytokines. So cytokines are immune molecules that play a very important role. Some of them can be in terms of like activating certain pathways that are necessary to maintain homeostasis, and as well as how we respond to viral pathogens or antimicrobial organisms in general. So one of the work that I'm looking at is IL-2. So IL-2 is a key immune cytokine that is very important for T cell development, but it's also very important in other contexts too. So right now, looking at what are the initial immediate responses to IL-2. So when you administer IL-2 and different complexes of IL-2, what genes are turned on, what genes are turned off, what cells are responding in the first place. So this will give us an idea of which pathways are very key in our responses, or which genes are very key in our responses to pathogens. And also IL-2, it's also important to, this is the context I'm looking at it from, but IL-2 is also very important in terms of like cancer and immunotherapy, but that is another field. Yeah.
So I think that's a really interesting study. So trying to understand how cytokines, the responses that follow the release or activation of certain cytokines in the immune system, right? Yeah. So
Right.
so I don't know how much you've done yet, because, or what you look forward to, maybe we should just delve a bit more into the field and the research of trying to understand how, the genes that are expressed when cytokines are secreted, because I know from what you're saying, the field already overlaps immunology, genetics, molecular biology, and all of that. So it's kind of multi-layered. So let's delve more into that space and get more context to the findings you are exploring, or the questions you are exploring, and the possible findings or implications you think they would have in the study of diseases, maybe infectious diseases in particular. Yeah.
Right. So in terms of, so right now we've published one of the work already. So it's on the common gamma chain cytokine, so the IL-2 family, as I mentioned. Usually this, yes, we know that these genes are from a family, but usually we may think of them as like one gene, cytokine just impacting like one particular cell type. But what we are seeing there is that these cell type, this stimulation of IL-2 with IL-2 can have direct impact on certain cells, but then it can also have indirect impact on cells, wherein when you administer it, yes, it can activate one cell. And when that one cell is activated, the cytokines that are released from that cell also influence like other cell types. So that is one implication. So now we are getting a broader understanding of that. And then we are also seeing that as is also common in very biological system, is that the cytokines can have similar responses. So then that means now that when people are thinking about immunotherapy, yes, they can target one particular cell, cytokines, but then they can also like complex it with another cytokines, and how that will boost the immune response. Because usually sometimes with IL-2, what is been seen is that sometimes it can have like a lot of toxicity effect, but now how can we modulate it to make sure that we are getting like very key specific responses? So that is what I'm doing, but the project in general is ImGen, so the Immunological Genome Project. And it is a consortium of immunologists and computational biologists that are mapping out the gene expression profile and the immune system. And the beautiful thing is that this data is made available to the general public. So if you're a researcher somewhere and do not have access to like RNA sequencing or looking at ataxic and how the chromatin are modulated, you can go to the website, look at these sequences and see how that can be applicable to the context of your research. Yeah, so I'll stop there for now.
Yeah. Yeah, thank you. So I think just quickly the website of IMGen website, I think you just mention it quickly for someone who would be interested.
Right. So it is immgen.org. So imgen.org. And there you would find a lot of sequencing data, different cell types, different contexts. So IL-2 was just one that I mentioned, but there are data on like interferon, gamma, the TNF, other cytokines, and then other contexts too. So it is very, very, it's a very resourceful website for researchers, especially if you're in low and middle income country and do not have access to these very expensive techniques, but you can utilize it and have answers within the context of your own research.
Yeah, thank you very much. So I think if I get the core of the research, you're basically trying to understand the let me say the mechanisms that or I think you're trying to understand the mechanisms that give rise to some of the responses that IL-2 can trigger, that's interleukin-2 can trigger. So when you understand the basis of those mechanisms, you are able to tweak the outcome. So if you understand, okay, maybe these are the genes or these are the cell types that are triggered, or these are the cytokines that are triggered in the cascade, you are able to kind of tailor the responses so that you can have less harm on the individuals who are maybe undergoing therapy that overlaps, that involves IL-2. Is that correct?
Yes. So that is one implication. So now you
Okay.
know the pathways, you know what genes are turned off, what genes are repressed, and also like what cell types. So with this, you can particularly geared like treatment or like dosing or even like complexing it with a different mutant or something. Which particular cell type do you want? How can you modulate that response? Yeah.
Yeah. Thank you. Thank you so much for that explanation. So is there something else about your research that you want to highlight? So before we proceed to the other questions, maybe like I think I often ask sometimes, maybe in the context of the African continent or in the context of infectious diseases that affect Africa, because I know sometimes some of these cytokines, they are sometimes better expressed in certain diseases than in other diseases. So like are there diseases that this finding will, or this work will have maybe direct or indirect relevance for in the African setting?
So, well, cytokines are very key in like infectious diseases, autoimmune diseases, cancer. So, like for instance, we saw with COVID, even though initially people were calling it cytokine storm, but you can see that with several infectious diseases, you have these inflammatory cytokines that are released to kind of signal in like fighting the infection. So the implication is like huge in several infectious diseases. So understanding their induction and also repression is very key, because we saw people used to call it cytokine storm, but it was one of the key cytokine that was implicated was IL-2. So for instance, in COVID-19, we saw that it was people that tend to have severe outcomes had more inflammatory cytokines that are produced, particularly IL-6. So then when we understood that, so you can see how you are able to kind of see who is going to proceed to like a more severe outcome or not, or how you can tailor a particular cytokine pathway to like mitigate the disease. So COVID was a great example for that too, but it's also very applicable in other infectious diseases.
Yeah, thank you. Thank you so much. So I think we'll proceed move away a bit from research and, try to explore your life as a graduate student. So some of your experiences and some of your perspectives and your life general as a graduate student. So I think the first question I would ask is how you coped or what was your experience dealing with failure and successes like? So how did you manage successes and failures when they hit you?
That is an important life question. And I feel yeah, if someone can navigate both ends successfully, then they will be a well-grounded individual. So I'm still working on that. But some of the things that usually help, like for instance, whenever I have rejection, which it is very common in science, experiments don't work, right? And it takes several attempts to get them to work.
Yeah.
And then maybe also when I was applying after college, like for jobs and everything, there were places that rejected me. I would say it's knowing that one, who you are in those moments, because seeing the rejection as the rejection and not seeing that that is a failure as a
Mm-hmm.
person, like trying to, I would say, separate the experience from who you are and seeing that a rejection can be a redirection. Maybe, yes, you may be rejected in one place, but then that would cause you to look at another place, and that will open like a whole field of research for you too. And then also having people in your corner too that remind you of who you are and some of the things that you've done, some of the work that you've done, how you've been diligent and hardworking and meticulous in those contexts. So having good people, like a very strong support system. And in terms of success too, is that whenever they come, knowing that, okay, yes, this is one milestone or one accomplishment gained, but then there are several more to go. This is not the end. This is just another thing on the way to the journey, right? And seeing success really, I would say, like a journey and not necessarily like a destination, because when
Mm.
you see it as a destination, once you get what you want, then you'll be stagnant. But if you see it as a journey or some things that you accomplish along the line, then you keep going. Even when you have them, you see them as an encouragement. You see them like, okay, this is good. At least I'm in the right track. I'm doing something good. Let's keep going. So those are some of the ways.
Yeah, yeah. Thank you for sharing. And I think you highlighted a few personal experiences in when you were talking about the failures or dealing with rejections. And I think, like you said, it's very important that we do not, allow our negative experiences to determine our identity, to change who we are, what we think of ourselves. And like you also mentioned, getting support systems are also always helpful to navigate some of those difficult times. So I think another question I want to ask is, what are some of your favorite experiments to run? Maybe the typical experiments you run in the lab, and which of them are your favorites? Because I know certain experiments. Okay, let me just cite an example. For example, an experiment that takes several hours to complete. If it doesn't work out well, I know it will stink differently from an experiment that you could just complete in a few minutes. So which were your favorite or least favorite experiments to run in the lab?
Yeah, okay. So I would share some of my favorite. I would say it's after like isolating a particular organ, harvesting a particular organ, isolating cells, different cells to like flow cytometry, and then doing RNA-seq and getting the data back. Because like once you get the data back, it's like you're looking at tens of thousands of genes, right? So for instance, like in the mouse, like around 53,000. And so that is like for me there, the possibilities are endless. It's like, okay, which pathways are turned on? So the excitement begins, wherein when you're looking at the data, you're like, okay, in this context, maybe it's this cluster of like genes, but in this context, it's different. So having that excitement of those different types of genes, it's like very rewarding. And I would say some of my least favorite, what could that be? Well, it could also be flow cytometry, wherein you're trying to look at some rare population or even like sort those rare population. One of my least favorite back then to do was gamma delta T cells and sorting like, let's say, 10,000 of those. It's like hard in some context. And looking at like you're sitting there looking at the thing going through the flow cytometry sorter, if it's like fax area, and just like, yep, you're sitting looking at the screen and waiting till you can get the particular number that you want. So yeah, that can be challenging. But then usually I'll bring either like a paper or listen to a podcast during that time. So that made it like better. Yeah.
So I think you've already act your way into coping with some of the difficult or not so interesting
Oh,
experiments. Yeah,
right.
yeah. Yeah, so
Yeah.
I think for
Mm-hmm.
those who are listening, so if there are experiments you don't really enjoy running in the lab, just find a way. Maybe listen to a podcast, listen to music, or
But
just do something alongside. So you can get
right.
pleasure or get your kind of find pleasure in something else while you're going through that difficult time. Yeah. So
Mm-hmm.
yeah.
Yeah.
So yeah, thanks for sharing all of those. I think you were mentioning some kind of terminologies, but I know would cope the audience would cope like gamma delta T cells. Like I think from your description, they are pretty it's very small class or kind of quite not so common as other cells. So it's quite difficult to get the exact number you need on time compared to maybe other cells that have a higher population, right?
Yeah, right. So it's more, yeah, it's the percentages. So for instance, if you're sorting, they're a subset of like T cells. So like if you're just sorting T cells based on like the major markers, like whether they are CD4 or CD8 positive T cells, those are very fast. Like the moment you just put the population and then start collecting, it's like you can get those within like seconds to a minute, like huge percentages, because they're huge. But with gamma delta T cells, they're like like smaller percentages of T cells. So it can take a while to get like tens of thousands of
Yeah.
those. But I do recommend podcasts, and it's great that if you're listening to this podcast right now, if you're not doing your experiment, but if you're going through a challenging experiment, please listen. It'll help.
Yeah, sure. Yeah, thanks. Yeah. So I think still on your experience, but not just focus on grasping. I think we'll now kind of um focus on your journey and your life in general. So I think the question I want to ask are the principles that guide you, your perspectives, some of the guiding principles and perspectives you have. And I think I will just pair it with this question about your journey so far and your
Mm-hmm.
experiences. So how maybe highlights of those principles in your experiences so far. So these
Mm-hmm.
my experiences, my journey so far as a young girl from Sierra Leone who kind of topped the kind of was the best student, best female student for the WAS exam in your country, and then getting to this pedestal you are now. So what are some of those things that you think have guided you or kind of shaped your life or your career, and that someone else who aspires to be like Kumba someday, maybe preparing for WAS exam too, or in undergraduate institution, how they can also learn from that experience and also build their life.
Yeah, thank you for that question. So I'd say looking back, some of the challenges and that I've gone through, and getting to where I am, the pathway in getting to where I am today, some of the principles that have guided me one of them would be perseverance, because knowing that nothing comes easily, success doesn't come easily. You have to put in the hard work, you have to be diligent, and especially as you go through life, there are several challenges that would come, several life happens, several things that will come through your pathway. So for instance, I mentioned that Sierra Leone is a war-torn country. We experienced civil war, we experienced the Ebola outbreak, we experienced the mudslide, and during these things, like I can know people that are affected, and so these things can go through, can get us down, but you need to persevere. And also personally for me, one of the challenges have been like losing my dad when I was in undergrad, and my dad and I we were best of friends. And so then it is in those moments, like for instance, that you need to keep going, because it's going to be challenging. It could also be you being rejected or you not getting the WAS results. Yes, I got my WAS results in my first sitting, but then even if I had not gotten my WAS results in my first sitting of the exam, taking the exam, like having that push, that resilient spirit to take it again. So that is one. Success doesn't come easily. And then I would say the other thing is pursue excellence. Like whatever you do, make sure that you are giving it your all. Make sure that you want to improve. And when I mean excellence, it's like always that mindset of like, now I have it at this standard, but next time when I'm doing it, or the next thing I'm doing, I want an improved standard. I want it a little bit higher. I want it a little bit better. So always having that excellence mind spirit, it will really take you far. It will take you to places wherein you would not dream of, because people will get to see that work ethic in you. People will get to see the work, the excellent work that you're doing. They will get to respect you for that, but then also avenues will be open for you to share those work or your experience, so that at least that could better humanity. So having that excellent spirit. And I'm not saying be perfect. I'm not the one to say I want to be perfect. I slightly have a better way to frame that, which is pursuance of excellence.
Excellence, yeah.
Yeah. Whereas being perfect, it's like, oh, okay, everything should be like spotless. No, life is going to happen. Like for instance, I shared with you that during undergrad, I lost my dad. That affected my grades. It affected my grades severely in college. But then having that excellent mind spirit, it's like, okay, I'm going to do better. Yes, this is a bump in the road, but I am going to try. So having that excellent spirit. And then I think another part too that have guided me is service, like giving back to my community. And yes, I don't mean in like very grandiose or big ways. Yes, I've had the opportunities to do that through like renovation of orphanage home in Sierra Leone and everything. But service, it's the little things, it's the everyday thing. For instance, it could be you knowing a particular subject. Let's say, for instance, you know further mathematics, or you know a neuroscience subject, or you know microbiology more than someone else, a concept that you understand more than someone else. You can share with them, you can explain the concept with them. And always having that ability to want to help humanity, want to help people around you, giving back to your community, because it's only when you can really build people around you, build your community. That is what true success is about. It's about that ability to give back. So I would say those are the three for now that have been very key in my development.
Yeah, thank you. Thank you. So perseverance, excellence and service. So I think just to mention that, I think one thing that stood out for me when you were speaking is the fact that you mentioned that even though you don't get the results you want, that shouldn't keep you from trying again. Like you highlighted that although you got the best worst result in doing when you took the first exam, but then even if you hadn't gotten that best, or you hadn't even made all your papers, you would have still tried again. And in that second try, you would have still given your best. So I think that's really important, that at every stage of our life, whether things are working out as expected or no, things are just, life is just hitting us left, right, front, back, and center, we should make sure that we are bringing our best foot forward and giving our best. Yeah. And then we'd always get the rewards. Maybe not now, but someday the rewards will always come. They would always find us.
Exactly.
Yeah, thanks. Yeah, so I think the one question I wanted to ask is about immunology in the continent of Africa and about science generally in Africa. I think sometimes when I think about some of this, our experience in Africa, more recently, I think I've been having some kind of excitement, because I think a couple of things are happening gradually. There's like attention are being given to certain things now, unlike before. Of course, there are still several challenges, but as in your role as an immunologist, as a graduate student who has had this exposure, what to you are some of the things that the continent needs to give attention to, and what are areas that you think we can still do better? And perhaps not just telling us what we can do better, but how, if you had suggestions or thoughts on how we can actually go about getting the kind of results, placing Africa at that pedestal that we are capable of, because I think we have the potential, but things are just the way they are. So what are your thoughts on that? Thank you.
Right. That is an excellent point. I want to echo what you said and emphasize, begin from there. Like, we have the potential.
Yeah.
There are several brilliant young Africans that are capable of just asking the right scientific question, asking why, and even coming up with answers to that. There are excellent minds on the continent. And one thing I think that we can do is to continue encouraging one another, to keep that passion alive, to keep that desire for science alive. And one way we can do that is like us sharing our stories, like what you're doing. And this could even be to like the primary school level, wherein if there are students there that are interested in science, like encouraging them, being an example to them, mentoring them, so that that brilliant mind, that curious mind that passion could always be there. And then I think the other thing too is that could be done, could be creating the infrastructure. So like I mentioned, right, for instance, I'll give my country's example. Sierra Leone was war-torn, and several infrastructures back then when I was growing up were not there because we were coming from the war and rebuilding and all of that. And so just imagine if the infrastructure is there, like in several African countries, there to like support these minds, there to create the environment, have, for instance, the apparatus that are needed for the research. So because I remember back then, like utilizing one pipette for like a whole class, like using using one pipette for the whole class. Can you imagine? And sometimes I go back, now I have my own pipette, and now I have my own, like, not even one, like a set of of P20s and P10, like the whole set, having things like that, and going back to when we were using like one for the whole class. And sometimes it was even the mouth pipettes back then. So just imagine if we had the infrastructures in place to support these brilliant minds. I think the possibilities are endless. We'll be there researching the diseases that are affecting our continent, and also like giving back to the world, because Africans, we're capable of doing that. We have the minds. We also need the infrastructure.
Yeah, thank you. So I think I love how you structured the responses, starting with the human resources and then progressing to the infrastructure, because I think we first of all have to make sure that we are, nurturing the young people, nurturing the minds that are going to make use of that infrastructure. But of course, the infrastructure is also very, very important. Yeah, so thank you for sharing those perspectives. And I think just mention that yeah, of course, I think like with the kind of young minds we have now, of course, people I've seen like you, like other individuals who brought on the series of the podcast and several other young Africans who are doing a lot of things in sciences. I do have hope that the future of Africa will be much more better, much more glorious than what we are currently experiencing now. And we just hope that everything will just fall in place like we are hoping that they do in no time. So yeah, thanks so much for sharing your story, and thank you for being an example, an inspiring example to several young girls and young people from the continent, and yeah, I think across the world too. Thank you so much. Before we take the last question, which is like a fun random question I'm going to ask you. So I'm just going to give you like 1-2 minutes to just say any other thing on your mind that you have not had the chance to say. Maybe something on your mind. I'm not going to cite examples, but any other thing you want to just
Right.
highlight. Yeah. Thank you.
Yeah. So one thing that I would want to highlight is one for young scientists on the continent to really utilize the internet well. There are several free online courses from renowned institutions around the world. I want people to make advantages of those. These can be through Coursera. Take those classes. So, for instance, if you're interested in computational biology, looking at bioinformatics and analyzing immunology sequence data, like go on these I would say platform, learn how to code, and there are free available like sequence data. So do that. Start practicing, start utilizing your free time. So that is one thing that I would say. And then also reach out to people, reach out to young Africans that are out there in different institutions. You never know. Maybe they may be open to like mentoring you, sharing their experience with you, and guiding you if you want to follow their footpath. And that would be like an excellent way to get out of there. So those are the two things that come to mind right now.
Yeah, I think those are very, very great advice, pieces of advice, right? Using Google, learning how to navigate the internet to find resources, and also knowing how to reach out to people, to knock on doors and see if anyone would open and respond to you. So I think those are very, very important points that young persons from Africa, in fact, not just young persons, but individuals can use to, just make progress in their career. So the fun question I'm going to ask next is for you to a fun request rather, not a question. So I would like you to share one of those eureka moments you've had in the lab. So like maybe something that you've been working on that wasn't giving you what you wanted, but then you had that moment and you're like, oh, like don't maybe running about and just shouting, Eureka, Eureka, Eureka. So let's have one of those.
Yeah, let's see. There are moments like that, both in terms of like the computation, analyzing like the sequencing profile of the different immune cell types, where in a particular code might not be working, and like spending like hours trying to debug it and finally doing something and then it works. But then there is also the experimental component of it. So looking at like flow cytometry and trying to like separate like different like cell types, right? So there was, in this particular panel, it was like 14 cell sets panel. So looking at 14 different immune cell types. And for one of the panel, there is the red pulp macrophage. So it's a particular type of macrophage, and trying to like get the separation clearly, wherein you can distinctively say these are the cell types. So like having having the voltages correct in the machine, right? And then also like the prep and how long you're staying, like optimizing all of those part. It took like, for that particular panel, all the other panels, the 3 other panels were fine, but for that one panel, it took like I think 3 additional weeks to get it right. And then there was the one day that it worked. I could see like the separation to the way that I wanted it, and I was like, yes. And yes, and that was one of the moments in my very earlier journey in immunology, experience with flow cytometry, that I would always look back on with fond memories. Yeah, but back then it was very difficult.
Yeah, yeah. I think one of those moments, right? So, yeah, I think of course it worked, and I think you already shared about some of the challenges that come with running all of these experiments. So, being a scientist is not all wearing lab coats and posing in pictures. It's a lot of sweat and investment. I know there is also the part where you just get tired and you're not even sure what next to do. So yeah, so well done. I think scientists are doing a whole lot, and this is just a tip of the iceberg to some of the difficult times they go through to just get the kind of answers that end up providing succor to some of the challenges that people are going through in the world. So thank you, and thank you for persevering till you got the answer that you were looking for.
Right. Well, thank you. Thank you once again for the opportunity to share my story, and thank you for just the amazing work that Immuno Africa is doing.
I like to think of Kumba's journey and research as being tightly intertwined. One obvious link between them is how her experiences spurred her to continue pursuing her childhood dreams of tackling infectious diseases. However, it's also remarkable that they both offer insights for making the most of resources, whether vaccines or immunotherapy, opportunities or challenges at our disposal. I hope you were inspired. Be on the lookout for our year in review episode that captures some of the most interesting highlights from this series and other episodes published in 2023.
Thank you for listening to this episode on Immuno Africa. If you enjoyed it, please feel free to share it with your network. You will learn first about future episodes and get other immunology related updates by following Immuno Africa _ other Immunology in Africa podcast on social media.